Hello, everyone, and welcome to today's live broadcast, Vaccines for the Treatment and Prevention of Cancer, presented by Dr. Keith Knutson. I am Tracy Aston Martin of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Bio-Techne. For more information on our sponsor, please visit biotechne.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Keith Knutson, a professor of immunology, Mayo Clinic. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Knutson, you may now begin your presentation.
Thank you so much, and thank you for the opportunity to speak with you all today about our vaccine program that we've been developing over the last many years now, and predominantly for breast and ovarian cancers, which is I'm going to talk about. But many of the principles that we've established and others have established can potentially work well for other cancers as well. Just a few conflicts of interest. We've licensed some of the technologies to a variety of companies who are moving these therapeutics forward. So let me just remind many of you that worldwide we have an extensive burden of breast and ovarian cancers, approximately 2.2 million cases per year worldwide and 800,000 deaths. So in the U.S., we have approximately 240,000 cases of breast cancer, about 300,000 total if you add in ductal carcinoma in situ, which is a very early form of breast cancer. And something that we haven't done much is the mortality rate, which stands at about 40,000 deaths per year. A much rarer cancer is ovarian cancer. We only have 22,000 cases. But because ovarian cancer is typically found in its very advanced stages, its mortality rate is quite high, with about 14,000 deaths per year. It's a very costly disease in the U.S. We estimate that it costs about $180 billion a year to manage all of the different aspects of breast cancer and ovarian cancers, which is about 0.2% of our gross domestic product. So we need to continue efforts to not only reduce the mortality, but reduce the burden that breast and ovarian cancers put on our society. So many of you may have a background in immunology. Some of you may not, and so I'm going to just give a little short course on the adaptive immune system. And just to put it simply, the adaptive immune system is the body's drug-making machinery, and it consists of predominantly three types of adaptive immune cells: the B cell, the CD4 T cell, and the CD8 T cell. So we typically think of CD4 cells as being helper cells. They coordinate the immune response. They're at high levels in peripheral blood. They can sense out antigen, and they can induce a wide variety of immune responses, including activation of cytotoxic T cells, which are cells that essentially lyse infected cells or tumor cells through presentation of antigen in the context of MHC class 1 molecules, which is shown here. CD4 T cells activate in response to antigen presented in MHC, typically MHC class 2 molecules, while B cells produce antibodies which can impact tumors in a number of different ways. And so there's about 10 to the 18th different specificities due to genetic recombination in the B cell and T cell compartment of proteins that can recognize specific antigens. So we have many, many vaccines, about 40 vaccines that are targeting about 28 different diseases here in the U.S. And so, literally, we've only touched the tip of the iceberg in terms of the immune system and how we can interrogate and activate the immune system against specific diseases, and that includes cancer. Now, what we've known over the past many, many years, and something that'll help you understand some of our later slides, is that in addition to the different adaptive immune effectors, as well as the antibodies or the T cell receptors that recognize MHC molecules, there's many different flavors of the adaptive immune system. We typically think of, based on earlier studies, that the immune system either polarizes into a Th1 or a Th2 profile. And that has been a long-standing paradigm that has persisted in the immunology literature for many, many years. But over the past decade or so, we have seen many descriptions of other types of T cells. And these different flavors of T cells are becoming essentially associated with a variety of different types of diseases, and some get activated in specific types of cancers. And so as we move forward in vaccine development, it is going to become useful information for us to try to understand what types of immune effectors to activate, and I'll come back in more detail on that a little bit later. So we've known for both breast and ovarian cancer for many, many years now, since the mid-1990s, that the immune system naturally responds to these cancers, and that the T cell responses are associated with improved survival, telling us that the immune system can have activity. The question is, how do we develop therapeutic approaches to target these cancers based on vaccine technology? This is just an example of a study that was published way back in 2013, looking at gene signatures associated with a couple of different flavors of T cells. Th1, which typically is like a macrophage associated immune response with high levels of interferon gamma, lower levels of antibody responses. But what we see here in HER2 positive breast cancer is that there's follicular helper cells also associated with improved survival. So the higher level of gene signature that you have in tumor, the better survival that you have relative to not having that signature. So this is just an example of the signatures of immune responses that we typically see within cancers, demonstrating that the immune system can have activity. And the goal of vaccination, the goal of immune checkpoint blockade, adoptive T cell therapy, is to harness that incredible activity to eliminate the tumor or even prevent it. Part of the work that we did early on was initially understanding what the immune system was responding to. And so we did a number of studies early on to demonstrate conclusively that while immune infiltration is associated with improved survival, these patients also have peripheral levels of B cells and T cells that are specific for specific antigens that are overexpressed in the tumor microenvironment. And so we continue to put together information from breast and ovarian cancers, and others do this with other cancers as well, to try to identify what those targets are that we can formulate into cancer vaccines. So one way to think about it is to divide them up into three groups, basically. So we have microbial neoantigens, we have mutation-derived neoantigens, which are very popular these days to interrogate and formulate into different therapeutic strategies. And the approach that we took several years ago was to develop vaccines based on non-mutated self-antigens by virtue of overexpression or other abnormal presentation in the tumor microenvironment, revealing what we call subdominant neoantigens. We know in breast and ovarian cancers that there's no microbial etiologic agents that we can target. For example, in cervical cancer, head and neck cancer, or in liver cancer, we see viral elements, so human papillomavirus for cervical cancer and hepatitis B virus for liver cancer, and we can target those with antigens. Now that we have good sequencing technology, we can understand the landscape of mutation-derived neoantigens. And I'll get a little bit into that as time moves on. But most of our efforts have been put forth in identifying non-mutated subdominant neoantigens that we can formulate into vaccine approaches. And this is put graphically in this particular slide, where on the left side you can see a normal cell and you have a protein, for example, HER2-neu, which is often overexpressed in breast cancers, gastric cancers, esophageal cancers. Under normal conditions, it's expressed at very low levels, and you have epitopes that derive from the processing of those levels of HER2 expression that get presented dominantly in MHC molecules. And your immune system is good at tolerizing itself either through central deletion of the T cells or antibodies, or the generation of regulatory T cells. And I'm not going to go into all of the data that we have to support that, but this is something that we have to regularly show, for example, to the Food and Drug Administration, that the epitopes that we have aren't expressed on normal cells. Whereas in a tumor cell, when you have aberrant expression of, say, HER2-neu, for example, again, you have mislocalization in the cell, you have different degradation, you have different glycosylations that can all impact the presentation of epitopes. And we see this with a wide variety of tumor antigens. And what happens is, due to this overexpression, you get processing and presentation of what we call subdominant epitopes. And your immune system is not tolerant to those. And so your immune system recognizes those. And we can interrogate those, we can identify those, and formulate those into vaccines. And actually, we started this back in the mid-1990s when there was very little understanding of the potential role of vaccines in treating and preventing cancer. And so with this, we targeted HER2-neu at the same time that Herceptin was being developed, and we came up with several different vaccines, and we immunized individuals, many individuals with these vaccines. For example, HER2-ECD vaccine, the extracellular domain, the intracellular domain, and a couple of other different vaccines based on class 1 epitopes. Most of the focus of our work has been on MHC class 2 or CD4 T cell epitopes, given that we want to apply the vaccines as a prevention agent. So secondary prevention in individuals that have been previously treated for breast or ovarian cancers that had been rendered disease-free. And most of the vaccine studies that were done back at that time were done in individuals with very advanced stage disease. And so that was very problematic, because it turns out that in very advanced disease, when you have bulky malignancy on board, you have a lot of immune suppression that prevents the development of the immune response and prevents its activity in the tumor microenvironment. So we positioned ourselves very early on as vaccines as a strategy to prevent disease. And this is shown here back from a study that we did a long time ago, where we systematically identified epitopes from HER2-neu and gave those when there was no disease burden, generated that immune response, and we could show that we could block the development of tumor. Whereas if we started immunization after we gave a big bolus dose of tumor cells, it takes a long time for that immune response to develop. And during that time, it turns out, and we understand this more fully now, that the immune system actually develops immune suppression. So I'm not going to go into all of the mechanisms. There's several. we can talk about that during Q&A. For example, regulatory T cells is one group of cells which are known to infiltrate into breast and ovarian cancers and block immunity. In fact, we did a lot of work back in the early 2000s to show that patients that had high levels of regulatory T cells in their tumors had worse outcome because it blocks that anti-tumor immune response. So hence, our application of vaccines has been in the minimal disease setting. However, we now have agents that block this immune suppression, opening up new avenues for us to combine vaccines with other types of strategies, say, for example, immune checkpoint blockade, that will enable the immune system to have an impact even though there's bulky malignancy and immune suppressant in the tumor microenvironment. But at that time, when we were developing vaccines for HER2/neu, we had very limited bioinformatic technologies to help us. We had actually algorithms that were based on mouse MHC. It was successful, but with the development of new bioinformatic strategies, we're able to, over the last several years, predict self-antigen, overexpressed self-antigen, HER2/neu in this particular example, using advanced algorithms based on known binding to many of the MHC class II molecules that are present in the human population. And this is just an example of how we take predicted epitopes and we bind those after prediction to MHC molecules and then determine which ones have higher affinity for those MHC molecules. And we tend to prioritize those ones that bind to multiple different HLA-DR molecules in order that we can construct a degenerate epitope off-the-shelf vaccine for use in individuals with a prior diagnosis of breast and ovarian cancer. And then following that bioinformatic and that binding studies, and we go to humans and we actually ask if individuals have pre-existent immune responses, and we prioritize further the identification of those neoantigens with the observations that pre-existent immunity exists in these individuals. And so this is just an example of a peptide-based vaccine, HER2/neu P59, P88, 422, and 885, which we constructed. This particular vaccine as a pool binds to 95% of the HLA-DR molecules that are present in North Americans. And you can see the distribution. So 3 of those are extracellular and 1 of those is intracellular. In fact, we've advanced this particular vaccine significantly, which I'll get into. And it's been a useful tool also to interrogate responses to a wide variety of HER2/neu targeting agents like Herceptin or margetuximab or other agents which bind up HER2/neu, where we can see that when those patients are treated with Herceptin, they develop immune responses to these subdominant epitopes. So that gives us confidence that a T-cell repertoire exists in these populations that we can formulate into vaccines. So in this particular vaccine, those 4 epitopes, we just recently showed that individuals immunized with the 4 epitopes can rapidly develop immune responses to these epitopes. And in fact, the vast majority of individuals responded with the generation of immune responses. Nearly 100% demonstrated immunity to at least one of the epitopes or to the processed antigen, either ICD or ECD. And importantly, the immune responses were long-lived in individuals, and we could measure those out to a couple of years, even with just a short vaccination series. And so in this particular kind of abbreviated subset of that data that we now have published, we show that following immunization, we can go back to those patients at 2 years, 2.5 years, for example, and we can see that T cells specific for the epitopes remain elevated And this ideal outcome, of course, of a vaccine is to have persistent immunity that can provide protection. We also see that in those individuals that as a result of that T cell immunity, their HER2 antibodies, their endogenous antibodies increased. And in fact, what we showed in patients with either in the adjuvant setting or the metastatic setting, that higher levels of antibodies against HER2 are associated with improved outcomes. So this is telling us that we can vaccinate patients and improve survival following a diagnosis. And in fact, we've just started a phase 2 clinical trial, and that's going to be conducted in about 190 patients that have undergone treatment for HER2-positive breast cancer, neoadjuvant chemotherapy, and neoadjuvant trastuzumab-based therapy or HER2-targeted therapy that have not or that did not demonstrate a complete clinical response to neoadjuvant chemotherapy, had the remainder of their tumor surgically removed, and we're going to be in a placebo-controlled fashion and blinded, giving these individuals this vaccine, along with, in this particular case, TDM1, which is, for those that don't know, it's a conjugated form of Herceptin that carries in a drug to block tubulin activity. So the idea is that the delivery of vaccine boosts those immune responses that are induced during maintenance therapy in these individuals in the adjuvant setting and be able to protect them from relapse. And so we'll have a readout on this phase 2 clinical trial, which is funded by the Department of Defense in about 5 years or so. We've done similar things with other vaccine antigens over the course of these several years. Another example that we have is that we see that certain subsets of individuals with breast cancer, specifically those with triple-negative breast cancer, as well as ovarian cancer patients, have very high-level expression of the folate receptor alpha. This is a protein that is typically only expressed at very select spots, typically away from the immune system in the body. Cancers, for a variety of reasons, maybe the uptake of folate, overexpress, and this aids in growth of the tumors, allowing them to get ample supply of folate for their metabolism. So we went through a similar bioinformatics approach and identified 5 antigens, class 2 CD4 T cell activating epitopes from the folate receptor alpha. And a couple of years ago, actually published this in Clinical Cancer Research, in which we demonstrated that we could safely immunize individuals. And this looks at the vaccine period. You could see that individuals developed immune responses to the epitopes, to the folate receptor alpha protein. And when we went back to those patients between 12 and 18 months in follow-up, we could also see that they demonstrated immune responses. And this is even in the absence of any type of booster. So this approach is also safe in generating persistent immunity in individuals. This has gone on into a phase 2 clinical trial where we're asking the same question that we did in HER2-neu, but in triple-negative breast cancer, whether or not we can immunize and protect individuals from relapse. So this trial is ongoing. Multiple centers around the country are participating in this clinical trial, in which we delivered the vaccine after treatment of patients with triple-negative breast cancer, standard conventional treatment, surgery, and chemotherapy. Triple-negative breast cancer is a prime choice for this vaccine because relative to the other vaccine or the other cancers, it has much higher levels of folate receptor activity. But then during this time frame, we started to think about the types of flavors of immune response that were being detected in tumors that were being associated with these other subtypes of T cells. Rather than the Th1-Th2 dichotomy, we see in ovarian cancer, for example, that IL-17 and Th17 T cells were associated with improved survival. This is studies that were actually done by another group. but it emphasized to us that we needed to start thinking of ways to develop adjuvants for a vaccine that would stimulate these unique subsets of T cells. And so this shows that patients with high levels of IL-17, and this has been reproduced by numerous studies now, have much more improved survival in ovarian cancer relative to those that have lower levels of IL-17. And so our thought was, could we construct a vaccine that would specifically stimulate the development of a Th17 vaccine approach, and if we could mimic that in patients with ovarian cancer. And so we started a clinical trial, and what we do is we teamed up with Martin Cannon from the University of Arkansas, who had seen that if you block p38 MAP kinase in dendritic cells, that's shown here, you actually get a unique phenotype of dendritic cells that enable the generation of Th17 T cells. I'm not going to go into all of the data, but we used this and we showed that blockade of p38 MAP kinase results in lower levels of IL-12 and higher levels of interferon gamma, but at the same time secreting very high levels of cytokines that are linked to the development of Th17 T cell immunity. And we see that this approach is unique for generating Th17. So we've developed animal models to study this. And what we see is that these dendritic cell vaccines that are based on blockade of p38 MAP kinase and pulse with antigen not only activate Th1 immunity, as shown here, relative to conventional dendritic cells, but specifically and uniquely activate the development of Th17 immune effectors. So this particular vaccine enables the development of Th17, but also keeps Th1, Th2, and antibody immunity intact. And so we conducted a phase 1 clinical trial, which is actually submitted and is under revision, and that was positive. So we had, very interestingly, we had a subset of individuals, we're out at about 5 or 6 years now after initial diagnosis and treatment of these individuals that haven't had relapse. Now, we would have expected at this time probably on the order of about 5 to 10% of patients without relapse. So we have very durable protection of patients, the primary diagnosis of ovarian cancer that had been treated. And that, whether or not they progressed or not, was linked to the development of an immune response. So all of the patients developed immune responses, but those individuals that had higher levels of immunity, and particularly interferon immunity, they all developed IL-17. That wasn't associated with progression or no progression. And they had higher levels of antibodies that were induced, which was kind of a surprise to us. Against the folate receptor alpha, those that had no progression had higher levels. Immunity was persistent against all of these epitopes that we included in this, both interferon gamma as well as IL-17 positive T cells. Antibodies were also persistent as well. So this is telling us that in this particular population, IL-17 inducing vaccines may have activity, and it may be through the generation of a coordinated, broader spectrum immune response that potentially involves not only IL-17, but also interferon gamma, Th1 cells, as well as antibody responses. And so we've modeled this. We've developed a similar vaccine, and relative to conventional dendritic cells, shown that animals that have been injected with an aggressive form of ovarian cancer can be protected more so than those that are immunized with conventional vaccines. And in fact, what we think is happening is something very unique, is that Th17 is actually driving eosinophils and macrophages in the tumor microenvironment. The antibodies that get generated as a result of vaccination then bind to Fc receptors, and through antibody-dependent cellular cytotoxic or cellular phagocytic mechanisms tumors that are in the peritoneal microenvironment are killed. And as a result of that, these mice can live for a long period of time. And in fact, if we couple this with anti-PD-1, for example, we see very extended survival out well past a year in mice, making it very difficult to do these kinds of experiments because it takes so long. So this is advancing into a phase 2 clinical trial, which we're just getting up and running right now, in which we're going to immunize patients with autologous dendritic cell vaccines that have been treated with p38 MAP kinase inhibitor. And following their normal course of therapy, which is neoadjuvant chemotherapy sometimes, all of them get surgery and then followed with chemotherapy. They'll be randomized to one of two arms. We anticipate in this particular trial that we will vaccinate about 78 patients, or excuse me, enroll about 78 patients. 2/3 of those will get the vaccine, and then we'll give them boosters to continue their the development of their immune response, or augment their immune response or maintain it over the long period, and then we'll watch those individuals, see whether or not we can reduce relapse, and then try to understand where it's failing. What are the mechanisms by which the tumor relapses? There's a number of different possibilities. It could be PD-1, PD-L1, it could be antigen loss, it could be other types of immune suppressive mechanisms. But this is telling us that if we think about what types of immunity are activated in the tumor microenvironment naturally and what are associated with improved outcomes, that we can build our vaccines based on that knowledge to enhance survival in individuals previously diagnosed with cancer. Now, another way that people are thinking about using vaccine, including us, are to combine with immune checkpoint blockade. And what's of interest here is developing neoantigen-based vaccines, something that we're starting to work on and starting to do clinical trials as well. So this is a results of a study that were published now a few years back, in which we had a neoantigen vaccine, and I use that term kind of loosely. It was neoantigens derived from mutations as well as subdominant epitopes that we had identified. In fact, this was 3 epitopes that we had identified, C3, L3, and 1 from HER2, legionnaire, and beta-catenin. And as you can see, the vaccine, when given alone, can, for a very short period of time, just a week, can begin to induce regression, but then the tumors rapidly grow out, as you can see here. Now, one of what was of interest to us at that time was whether or not the combination of vaccine plus anti-PD-1 could result in sustained anti-tumor immune responses and eradicate the tumor completely. And in fact, we found that to be the case when we delivered simultaneously anti-PD-1 every several days with 3 injections of vaccine over the course of a week. And what we observed is that we had a marked synergistic interaction between vaccine and anti-PD-1. And in fact, 75% complete regression in this particular breast cancer model. Those tumors that grew out, we continue to analyze them, are highly immune edited. So they figured out ways to lose the antigen expression, despite the fact that we had 3 antigenic targets within the vaccine. But nonetheless, this is showing us that we can significantly improve survival of those individual mice that were treated with anti-PD-1 by the inclusion of vaccine. And so this has been, there's a couple of papers, an intervention that people have been thinking about in terms of improving the efficacy of immune checkpoint blockade, which is effective in some individuals, but in the vast majority, not really that effective. And so, like others, we have started to develop personalized vaccines. So we can, choose to develop vaccines based on overexpressed antigens, but now with the development of rapid high throughput bioinformatics pipelines, that enables us to interrogate a patient's tumor DNA and come up with a battery of neoantigens that we can formulate into vaccines and give along with immune checkpoint blockade. And in fact, this is work with my partner, Jan Osman, who's in bioinformatics here at Mayo Clinic in Florida, where she developed a neoantigen discovery pipeline using all of the latest technologies that were available and some that she created. And in fact, this one is a little superior to other bioinformatic discovery pipelines that have been developed for neoantigens in that it maximizes discovery of insertional deletion or indel neoantigens. Also includes the identification of those that result both from insertions and deletions, but also frame shifts that are induced by fusion proteins as well. And it minimizes cross-reactivity in that it goes back and it scans all of the aberrant transcripts against known proteins that are present in other parts of the body, so we can eliminate new neoantigens that arise in the tumor that may have cross-reactivity. But importantly, it also has optimized HLA genotyping involved, and this has been a big problem with these informatics pipelines, is that the identification of the relevant HLA molecules is deficient with some of the bioinformatic pipelines that are out there, but we've managed to solve that by tweaking several of the pipelines in order to give us the best identification of class 1 and class 2 molecules. We still are trying to figure out how to prioritize those. And we think of breast cancer and ovarian cancer typically, because of what's written in the literature, as being those types of tumors which have low levels of neoantigens. But in fact, that turns out not to be the case if you look at neoantigens that come from fusion proteins or come from small insertions, deletions that result in frame shifts. And in fact, if you just look at single nucleotide variants, this looks at 835 TCGA breast cancer patients at class 1 and class 2. You can see that relatively low level of neoantigens from this whole group. But if you add on those frame shift neoantigens that arise either from insertions, deletions, or fusion proteins, you can see that's where the vast majority of activity is. And in fact, it turns out that patients typically have a low level of neoantigens derived as a result of single nucleotide substitutions, whereas they have very high levels relative to those that result from fusions or insertions and deletions, on the order of greater than 1,000 per tumor, whereas with the single nucleotide variants, it's only perhaps 10 to maybe a couple of hundred. So we also wanted to examine during the development of this pipeline whether or not the neoantigens are private or public. So are there shared neoantigens that we could potentially target and hit subgroups? And we weren't surprised when we did this analysis that the vast majority of neoantigens, we had nearly 500,000 that we detected from the whole cohort of 835 TCGA patients. The vast majority of those, about 440,000, were only detected in a single individual. So this is telling us that the vast majority of these are private rather than public. But that doesn't mean that there's not a few shared ones. So if we look at some other, transcripts that were present in these tumors, we do find some shared neoepitopes. So for example, there's two, one from this protein DIXDC1 and PI3 kinase, frame shift antigen from DIXDC1 and single nucleotide variants from PI3 kinase. There was 18 neoepitopes, mostly class 2 neoepitopes, that we could see in greater than 42% of patients, so greater than 5% cohort. But this is probably too low to formulate into a vaccine. We didn't see anything better than this. But what this is telling us is that we need to craft ways to prioritize those neoantigens that are private and put those into humans and test whether or not we can elicit durable immune responses. So interestingly enough, when we looked across the different subtypes of breast cancer, as well as the different tumor stages, we see very little differences in the amount of neoantigens. So it was initially thought that triple-negative breast cancer, for example, the basal type of breast cancer would have more, but that turns out not to be the case. In fact, the only significant differences that we saw were with luminal A, which is PR-positive breast cancer, had significantly lower neoantigens than the rest of the subtypes. And if we looked across stage, it didn't really matter. Those neoantigens were present right from the beginning. And so this is a potentially useful tool, not only for advanced stage cancer patients, but also for individuals with early stage cancer as well. But most importantly, what we found, particularly with the single nucleotide variants in the indels, is that lower neoantigen load correlates with worse overall survival in these patients. Whether we looked at class 1 epitopes or class 2 epitopes, we could see that their risk of recurrence was higher if they had low levels. Now, that could mean a couple of different things. Either that they didn't have enough neoepitopes to stimulate an immune response, or the immune response immunoedited these tumors and got rid of all the neoepitopes. I favor the first hypothesis the most, in that it turns the neoantigens could potentially be immunogenic in and of themselves without any intervention, but that may be targets for neoantigen-based vaccines. And so, in fact, we teamed up with Transgene recently, and we're starting to develop our own strategies to test whether or not we could generate immunity to neoantigens. This is a phase 1 clinical trial that we just started in ovarian cancer patients that are going to be given first-line therapy, platinum-based chemotherapy, followed by surgery. And of course, it's wait and see with those particular patients. And during that wait and see period, we're going to select neoepitopes based on prioritization schemes that we can formulate into a vaccine, and at the time of asymptomatic relapse, elevated CA-125, so we can start to immunize those individuals to demonstrate safety, with the hope that we ultimately could go into the combination with anti-PD-1 in these patients. So just lastly now, what I want to talk about is our efforts working with the National Breast Cancer Coalition, and even more recently with the National Cancer Institute Division of Cancer Prevention, to come up with a prevention vaccine for breast cancer. It's a topic that we've been working on for many, many years, driven by this large advocacy organization, the National Breast Cancer Coalition, in which the concept is to devise and implement into the clinic, into mainstream healthcare, a prevention vaccine. So, of course, I already told you that we can't do that with a vaccine based on viruses or bacteria, because there's no evidence that there's any causative agent that we can target that's foreign, nor are there any neoantigens that are shared across the board that we could formulate into a vaccine. So we're taking this approach by evaluating the development of immunity, whether or not it can prevent breast cancer, and it's still in its early stages, by targeting several known overexpressed breast cancer antigens like HER2-neu, which I was just talking about. And this is particularly promising in light of the fact that the normal breast is a gland just like any other gland that is exposed to the outside, develops its own mucosal immune system. This is just an example of normal lobules that from normal healthy individuals stained with H&E, a pan-leukocyte marker CD45 or CD8 cytotoxic T cells or dendritic cells. And you can clearly see, and we see this across the board in most individuals, that they have an immune response or an immune system already associated with the gland. So our question that we're going to be asking over the next couple of years is whether or not we can specifically intervene and get immune vectors specific for tumor antigens trafficking into normal, healthy breast cancer. So while this is being patented, the antigens are embargoed. The National Cancer Institute is working with us on developing the vaccine. It's going to be based on both a plasmid-based and a modified vaccinia virus Ankara-based boost in individuals, lessons learned from infectious disease vaccines. These are all prepared, and we expect that over the next 6 to 8 months that we will have manufacturing done, and that we will be able to implement in individuals a clinical trial in which we test these. And so we've met with the FDA, and they have advised us to go into disease-free individuals with metastatic disease or in stage 3 patients that have no evidence of disease and immunize first to determine whether or not it's safe. And we can at that time interrogate normal breast tissue to ask questions about whether or not we get infiltration or surveillance with the vaccine-induced T cells and B cells. So let me just conclude that vaccines are probably best used when used alone, not for bulky malignancy. So many, many years ago, there was a number of vaccine trials that kind of gave a bad name to vaccines because they were ineffective with bulky malignancy. But we know that's because the immune system is blocked in the tumor microenvironment by a number of different mechanisms. So we have, the possibility of intervening in the disease-free period to boost that immunity. And so this is where we need to do clinical trials. Now, these are pretty hard to do because this isn't something that you immunize and can see the shrinkage of tumor. You have to actually wait for these individuals to relapse. So these are particularly challenging clinical trials that take, a lot of persistence and effort in immunizing these patients and continuing to follow those. But we have several clinical trials now in which we're doing that. We also know now that with advanced bioinformatics that we can take advantage and perhaps make more personalized vaccines. The challenges there, of course, are that we need to come up with systems in which we can rapidly encode those neoantigens and deliver those. So a number of companies, as well as our laboratory, are working on very rapid production strategies for making neoantigen-based vaccines that, based on information that we get from the DNA and RNA sequencing and the bioinformatics. And we also know that now that we can repolarize immune responses and take advice from what the body's natural immune system is already doing to prevent redevelopment of the tumors and skew the immune responses to those unique flavors that we see. You can do that with autologous DCs like we've shown, but certainly there's going to be improved adjuvants that can help us skew the immune responses as well. And we need to, also be thinking about, are neoantigen-based vaccines going to be useful in treating patients that don't have any response to monotherapy, mono-ICB therapy. And then ultimately, there is growing enthusiasm based on the safety profile and based on animal models that we can actually prevent the development of cancers in the first place. And there's a couple of different cancers that people are trying to do that with, and we're trying to lead the way with developing the first prevention vaccine for breast cancer in association with the National Cancer Institute and the National Breast Cancer Coalition. So I'll end it there, and I thank you very much for your attention. Of course, there's just many, many people that have been involved in these. These are huge efforts by clinicians, by scientists, by advocacy organizations, granting foundations and organizations that provide monetary support to get these programs up and going. And the hope is someday that we'll learn better how to use vaccines to prevent disease. And there's been a number of different clinical trials that have been published in the past few years that show the possibility of, or the right clinical setting for vaccines in preventing or potentially even cancer in individuals. And with that, I will stop and take any questions that you may have.
Thank you, Dr. Knutson, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. We have a lot of questions coming in. Our first question is, can we produce cancer vaccine from secondary metabolites produced by microbes?
Well, I guess there's a couple of ways to think about that. We certainly, one of the issues, as I had alluded to with vaccines, is that we need to come up with better ways to skew the immune response. And so it could be that this particular question is referring to metabolites that skew the immune system. And so we could possibly, we're working on some of those. So for example, we're working on the development of novel lipids from some microbes like P. gingivalis that will enable the activation of the immune response in association with vaccine antigens. And so we could use those metabolites to activate the immune response and send it down one flavor versus another flavor, so to speak.
Do you know of anywhere or anyone that is doing vaccine development for metastatic colon cancer?
Yeah, so the place to check so there are vaccines that are or have been developed in the past, and the best way that you can check is to go to clinicaltrials.gov and type in colon cancer and vaccines. The problem with metastatic disease, of course, is that it's very heterogeneous, and the idea with vaccines would be to get in as early as possible during the development of the tumor. But certainly there are some viral-based approaches that I've seen out there, and perhaps these will work in combination with like immune checkpoint blockade or something like that. But you should be able to find ongoing trials with those on clinicaltrials.gov by typing in keywords that could help you find those.
Why did you target neoantigens to MHC class II HLA-DR instead of MHC class I to activate TC?
So our idea at that time, and it still continues to be very promising, is that we can activate CD4 T cells, and they can orchestrate the immune response. And so, and also in vaccinating during the period where there's no evidence of disease, the vast majority of cells are CD4 T cells, and they're very important in immune surveillance. And so the idea is, under the right conditions, they can respond to antigen that is draining into the lymph node and activate the immune response. And in fact, we find that to be the case in patients with ovarian cancer with the Th17 vaccine that I was telling you, is that we see evidence of vaccine-induced immune responses, but in those individuals that relapse, we also see evidence that their immune response is activated during the course of development. So it's a matter of immune surveillance and the fact that the CD4 T cells are central coordinators of the immune response, and they could potentially activate cytotoxic T cells if they sense the antigen. So that is the primary reason that we did that. And also at that time, we had done several studies in older individuals with ovarian cancer, and it turns out that older people have, in some cases, not all cases, but a defective CD8 T cell compartment that is enriched in memory cells responsive to viruses. And so it tends to be it tends to respond, at least in some of the older patients that we did, CD8 or class 1 targeting vaccines don't respond that well, and they don't generate strong memory immunity as a result of the lack of response, whereas that's not the case with the class 2 vaccines.
Like other vaccines, would cancer vaccines prevent healthy people from developing cancers?
So we need to test that. So this year we are going to test this self-antigen vaccine that targets multiple breast cancer antigens. In fact, the idea is to target antigens across the board of breast cancers and to prevent that. So this is just a brand new area, and in fact, I've been working on it for years, and it's something that hasn't been that well embraced. But I think with the the data that we have now, that you can generate immune responses to self-antigens, and the immune response can know what to do with those immune responses, that we can now go into healthy individuals. And the NCI Prevent program, or Division of Cancer Prevention at the National Cancer Institute, is looking into funding these types of strategies. In fact, we just received word of funding for the breast cancer primary prevention vaccine, which we hope to get into normal healthy individuals at high risk for developing breast cancer, but are otherwise healthy and have not had cancer in the past.
Thank you, Dr. Knutson. Okay, we have time for one more question today. And our final question is in reference to the immune editing. Is this on the tumor cells, immune cells, or alterations in both?
So that's a very good question. So it's a wide variety of different types of immune editing. So, and it just all depends on either the model or if you're in the human clinical setting. And so we see loss of antigen in some of our models where we immunoselect for tumors that don't have the antigen anymore. In the case of our Th17 vaccine, we see that all of the individuals that relapse have upregulated on their cell surface a molecule called PD-L1, indicating that they're modulating the immune system by blocking it. We also have, in ovarian cancer, seen that tumors can cause upregulation of immune regulatory molecules and suppress those upregulate immune regulatory molecules and suppress the cells on which they're expressed. And so it's variable, but I think the important thing here is that there are so many mechanisms by which the immune system is regulated. It has to be, otherwise it would kill you, that the tumors can take advantage of. And so close monitoring of the immune responses, as well as the tumor following immune-based therapies, I think is going to be essential for trying to identify the best use of it and how to, prevent a lack of response, so to speak.
Thank you again. Dr. Knutson, do you have any final comments for our audience?
Well, thank you for your attention. This is a very exciting time for immune-based approaches. Back in the 1990s, when we started working on it, when I was just a young postdoc, I remember that we were kind of outcasts, because there was no focus on that, and a lot of people didn't feel that it, really would work at all, because the immune system is for microbes, viruses, and bacteria, and not for tumors. But persistence in following the data, I think, is really what paid off here. And we're still in the infancy. We're using kind of crude immune regulatory procedures for altering the immune system, like anti-PD-1. But I think we're learning more and more, and we'll be able to make targeted approaches in 10 to 20 years by continuing to adapt to the knowledge that we gain from ongoing studies in tumor and infectious disease immunobiology. Thank you.
Before we go, I'd like to thank our audience for joining us today and for their interesting questions. We had a lot of questions today, and questions that we did not have time for, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank you, Dr. Knutson, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Bio-Techne, for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye, everyone.